Great. So we've got a really great session planned today, starting out with a double header between me and Alexa Warwick, who are both working on this project that we're calling yEvo, which is a yeast experimental evolution and genomics teaching lab, which has really enjoyed a lot of great support with Beacon. So thank you for that. And so what I'll do is I'll introduce the lab and talk a little bit about our first successful application of it. And then Alexa will take over and talk about some of the evaluations that we've done to make sure that we're doing a good job. And while, I've put here our website and our Twitter feed, and this is really the effort of a cast of thousands. We have, a bunch of students and teachers, multiple academic collaborators, and a whole group of people. And so I'll try to acknowledge people as I go, but, I just want to first upfront acknowledge that that's the case. Okay, so this all started when Ryan here emailed me, and thank you again also to Beacon for letting us invite people from outside Beacon Central. Ryan's actually participating in the Congress, and he and Bryce Taylor are going to host a sandbox about teaching experimental evolution on Friday. But Ryan emailed me, and that's what started this all. So he was really interested in seeing if he could turn his AP Biology high school classroom into a research lab. And he was familiar with the LTEE. And here is something that I have to thank Rich for, which is not choosing a eukaryote to start with, because then Ryan emailed me instead of emailing Rich. So the eukaryotic biology is more represented in the AP standards. And so Ryan was wondering, could we do experimental evolution, an experiment like the LTEE, but with yeast and over the shorter timescales, which would be compatible with a high school classroom. And what's really great is that, experimental evolution dovetails with so many of the different concepts that are taught in high school biology these days. There's evolution, obviously, because we have a genetics component to this, or genomics component, they learn about genetics. They can think about how aspects of cell biology change over the course of evolution. It all ties into metabolism, and then they can think about engineering the strains and learn some molecular biology. So it's really ended up being a curriculum that we've been able to intermingle with aspects of the entire year of biology coursework. So the sketch of the entire lab is kind of laid out here. And one nice thing about it is that it's kind of modular. So while Ryan has been able to integrate it over the whole year, other teachers have been more interested in taking just the evolution section or just the genetics section and talking about that. So the basic concept is that we do serial transfer with yeast, and we provide access to some sort of a stressor to provide a selective pressure on them. And as new mutations arise and take over, the students can see that their cultures begin to grow better. And at the end, and the end is defined in a few different ways, mostly practical, they send their evolved strains to us, and we sequence them. And then we send them back their a curated mutation list so that they can start to look through and start to think about, well, which mutations might be affecting my trait and why and how, and start to think about all sorts of evolution questions that way. There are other modules that can be added on depending on some of the results that they see. So for example, we found multiple genes involved in metabolism, and that those caused a phenotype that they could have tested for with the yeast. And so there's a metabolism module that got added on. We can talk about what fitness means and do competition assays. And that's an extra module. In fact, one of our teachers likes the competition module so much that she's involved her entire school with it. So there are different aspects of this that work differently well in different classrooms. And so helping to make this modular is part of kind of what part of the appeal is, because it's really flexible that way. Okay, so for our first time off the marks, we had to figure out how are we going to simplify experimental evolution so that we can be really successful in a high school environment. And in my lab, we do a lot of experimental evolution, and we often use continuous culture devices called chemostats, which are really complicated. And so we were like, okay, well, we're not going to have chemostats running in high school classes. How about if we do serial transfer, and instead of using pipettes and risking contamination, we just use individually wrapped sterile cotton swabs to do the daily transfers. And that has ended up working just astonishingly well. We've now implemented this in multiple classrooms with a variety of different selective pressures. But the one of the stars of the show has been what yeast we started with. And so these are pretty colors of yeast that are engineered in Jeff Buca's lab, and some of them, the orangey yellow ones, are affectionately called the carrot yeast that we made at Cold Spring Harbor yeast course in the summer. And these genes are the beta carotene biosynthesis pathway, genes from coral, genes from all sorts of things. And so by using these strains, the students kind of get a team identity. And we also are using this as a way of monitoring for contamination, and it gets used in the competition module that I'll tell you about later. The other thing is it's just fun, and the students get to think a little bit about some synthetic biology ideas, like, oh, how are we getting a yeast cell to manufacture, a high level of this coral pigment, right? And then the other piece of the puzzle is, what is the selection pressure? So, we've used a whole variety of things that I'll tell you about at the very end, but the one that we have done the most work on, and that has been just really successful, is this over-the-counter antifungal called Fungicure. And the things that are great about Fungicure are that, you can buy it at the store. It's safe. It's a 1 in 50,000 dilution of this stuff, kills lab yeast pretty effectively. And it's part of a family of antifungals called azoles. There's only a really limited number of antifungal families, and this is representative of one of them. So that means that we can take all the knowledge about the mode of action of antifungals, known resistance mutations to azoles, and the students can learn from that and also do some research on what they get in their new experiment to compare to what's already in the literature. Finally, we allow the students some agency in figuring out how much Fungicure to give their cells as the experiment progresses. So as they notice their cells growing better, they're given the option to up the dose. And we let them make those decisions. And sometimes, we allow them to go through extinction events every once in a while. We keep the backups in the fridge so that we can bring those back out in that event. But it makes for it kind of really to be a very clear demonstration that something's going on when now your cells are able to resist this higher dose that they weren't able to at the very beginning. So what did we find? So we've now run this lab for several years, and Paul Rowley got involved very early on and has a great team of students interacting with teachers in Idaho. And the data set that I'll tell you a little bit about is from sequencing 99 strains from 3 different high schools so far. And one thing that really appeals to me about the way that we've laid out our experiments here is that the data that we get is actually, really awesome data, in addition to being educationally interesting. And so we kind of think of this as like a win-win scenario where we're getting high schools to do evolution replicates for us and learn how to do experimental biology and think about genomic data. And at the same time, we're getting more replication than I could do in my own lab in some cases. So it's been a really fun project. So all of these evolved strains that we sequenced had at least one clearly causative drug resistance mutation, but those types of mutations spanned all sorts of different types of mutations. So already the students are starting to learn about mutation processes and genetics. And some of them were things like copy number changes. So we saw amplifications of the drug target. The azoles target ergosterol biosynthesis in yeast. So you can have them think about, oh, what happens when you make more copies of this gene that's being inhibited? We also got a lot of mutations that were gain-of-function mutations in activators of drug pumps. So if the drug gets into the cell, if you pump it out quickly, then you don't have to deal with it quite as much. And we saw this class of metabolic traits that's kind of a weird yeast thing where they can lose their mitochondrial functionality and become what's called a petite. So this one is fun because the students have to think about trade-offs, because these petites, as their name implies, make tiny colonies, but are actually more resistant to the drug. And so when they do the plating experiment and they see that they're tiny, but they're winning, it makes them really have to think about what fitness really means. Finally, we've done this enough now that we started to get a few new drug resistance genes, and we started to see patterns of mutation occurrence and co-occurrence and mutual exclusivity that have allowed us to discover some really neat epistatic relationships among some of these genes. And so we've gone into way more detail about this in a preprint that you can go read on bioRxiv. And it's been just really fun to have the students make some of these like real discoveries. And one of the students I remember in one of our interviews said, I feel like this is what a real scientist does. And indeed, it's true, right? So it's been really great. Getting back to that idea of fitness, one way that we have the students measure that is by doing a competition assay. And here's where the different colors really do come in handy, where we can mix them together, grow them in a concentration of fungicure that inhibits the ancestor really well, and then see by plating which one of them is winning. And we get really into this. We made a whole bracket and called it Yeast Madness, and the students get very into this. So this combination of like teamwork, where they're working with their partners to figure out how to evolve the best yeast, but then this competition, or they want to see, you know, whose yeast reigns supreme, is really fun. And the one of the teachers even got like other people in her school involved. They all made their own brackets and then followed the yeast battles to see who would win. And so once they do find out, you know, which yeast wins, they can start to think about, oh, this one has a mutation in a drug transporter, and so maybe that makes it better than this other strain that doesn't have that. And so it's a way of kind of trying to link some of these mutations to some of these phenotypes. Okay, so you know, this azole resistance implementation has ended up being really great and works very reproducibly, and also has this really great link to known biology, so that when students are like, oh, what's my mutation that I got? What is PDR1? And they type it into Google and it comes up with pleiotropic drug resistance 1. They go, ah, it worked, hooray. But we've been trying to expand conditions and also tailor them for different types of high schools. So a biotechnology classroom asked if we could do something with industrial relevance. And so, you know, at UW, we have a forestry department, and they gave us some wood waste hydrolysis that we evolved yeast to both resist the inhibitors that are present in that and eat the sugars in it more carefully. When the pandemic hit, and of course labs were closed, we built a kit that we wanted that to be as safe as possible. And so we used caffeine and we used baking strains, just in case, you know, somebody's kid's sister came over and drank their experiments accidentally or something. One of my undergrads, Margot Walson, piloted a lab on a different yeast phenotype of invasive growth, and that is very visual, and so that's been really fun to work on. And so there's just a whole range of this stuff. And so one thing that I wanted to welcome Beacon people to do is join us if you're interested in developing a lab for yourself and for your high schools in your neighborhood. Become a franchise. So you can then kind of tailor the selection conditions to what you're interested in and what your local teachers are interested in. And if you are interested in learning more about yEvo, we have a website, and that website has a contact form on it, or of course you can just email me. That's great too. We have a Twitter so that you can keep up with all the latest and greatest, where we post our papers. For example, our preprint on the mutations that I just mentioned, and we should be having a new preprint posted pretty soon on the pedagogy aspect. And I say stay tuned for that literally, because Alexa's going to talk all about that work up next. And then finally, I hope you'll come on Friday, because I think the idea of how you can use experimental evolution to teach many aspects of biology is a really fertile area. And so coming on Friday to brainstorm and think about this with a group of other people who all love experimental evolution and think about it really hard, I think it's going to be really fun. Okay, so with that, I just want to thank everyone. So here's Bryce here. So Bryce was a postdoc in my lab and is just starting as an assistant professor at Loras College. So he's going to move his franchise out there, and I'll keep some of the Seattle stuff going, and hopefully we'll be able to add more. And Margot and Angela, here's Angela here. She just started graduate school at Stanford Genetics. It's been really fun to have all these people involved. And to get to collaborate with one person I didn't mention is Fritz Roth and Atina Cole. They actually did some of the sequencing for us in the context of an undergraduate class. So we're hoping to kind of build this out even further to where some college level classes might be able to get involved as well. Okay, and with that, I will stop sharing and take questions. And I see something in the chat already. Great. Oh, bye, Rich. Are there other questions for me? You can just feel free to unmute and ask it, because we're a pretty small group. Okay, Anya's got one in the chat. Great. Recommendations for how to start these sorts of collaborations. Yes, that's a great question. So, you know, Ryan emailed me, right? So you probably don't want to just wait for some enterprising high school teacher to email you about it, right? So we have started to try to get the word out. You have to be a little careful about it because you want to make sure that you're able to deal with the bandwidth of supporting extra teachers who come on board. But we already, it turned out, in my department, we have another high school outreach program that is run through an NIH program, and they are really great in allowing me to come and visit. They have a board that is made up of a bunch of local high school teachers, and so they let me present this idea to them, and that was where we got a couple of connections and also some ideas for different things to do. And so we've just been kind of getting the word out through, not on a super systematic way, but other things that have happened is people who have heard me talk have their own kids who are in school, and they're like, oh, I wonder if you could get this going with my kids' school. And so we've gotten some personal connections that way. And not even just with scientists, but with like staff people that I know and things like that. So, it's very idiosyncratic how these kind of collaborations come to be. And but I think that the other thing that we are hoping to do is there's a science education partnership run by the Hutch here in Seattle, and they have a great network of teachers as well. And so if you can find some group that is already doing high school outreach at a professional level, then there's no point in reinventing the wheel or trying to develop that network yourself. But plugging into their network is really a great option if you can figure out who those people are. Thanks for the question. Anyone else? All right, we'll move on along to Alexa.
Awesome, thanks. I love when I don't have to redo all of the things and you can introduce my talk for me. So this has been a fun dual presentation for y'all, hopefully. So thanks for introducing yEvo. I'll be talking a bit more about evaluating it to see what kind of impact yEvo was having on the high school students involved here. And again, referencing the whole lab and the large group of people behind this, there's lots of co-authors on this particular talk, but many more who've been involved in teaching and helping with things along the way. So I was just going to back up briefly and ask that, well, why do these classroom research experiences? What is the importance of doing that? And there's a growing movement to incorporate classroom research experiences, certainly at the undergrad level with the CURES, the course-based undergraduate research experiences, might be a common acronym people are familiar with. And one of the reasons behind that is that there are a lot of benefits to students, to teachers, and to researchers that are involved with this. So I have just a couple of citations there, but I could list many more. Students usually show greater confidence and grasp of concepts and interest in STEM careers. And because these experiences are happening with all students, it helps to recruit and retain students that have been historically excluded and underrepresented in STEM and in science. Because those, again, those students are going to be able to be involved, and not just those that have special access to a research mentor, whether that be in high school or in undergrad. So teachers also get to incorporate these authentic science practices to meet their own teaching standards, and it can have a positive impact on teacher professional development as well to have these kinds of collaborations with researchers. And of course, the researchers get to fulfill broader impacts, which certainly for many of us, I think is beyond just a check a box, something we're really critically important and we're passionate about. And as Maitreya showed, you can collect data to advance your intellectual merit, your science goals. So it has a lot of benefits for all of the participants. And we're focusing today on microbial experimental evolution, which is particularly ideal for the classroom. Lots of different reasons. I'll just mention a couple things, like they can have relatively low resource requirements, so it could be incorporated into classrooms a lot more readily. Connecting concepts of evolution, cell biology, genetics, and then interests, sort of applications like medicine and biotechnology. And all of these cross-cutting concepts are emphasized within the next gen science standards that most states have adopted for K-12 teaching. So in addition to microbial experimental evolution being ideal, obviously we've selected yeast, and as already has been described, this is a popular species. It's attractive to work with for students for a variety of reasons. And you've already been introduced a bit to the general overview of the 5 modules within yEvo. These protocols differ a bit from some previous evolution teaching modules in that the experimental question is pretty open-ended. And those classroom projects, as Maitreya has already described, have a lot of agency for students to choose aspects of the experimental conditions, and they get to do competition, and there's lots of collaboration as they're trying to interpret their mutation data. So lots of things for students to be involved with. And just again, to emphasize that this has been a very collaborative endeavor with the idea stemming from a teacher, and certainly continuing to incorporate new teachers as we can as we move along. But really, I think the reason I was involved in the project was just to say, well, if we're doing all this work, what kind of changes are we seeing in students that are involved? So we focused on 3 areas and also included some demographic data as well. Those 3 areas of focus were some disciplinary content knowledge, mostly around evolution, and some just on basic, like, what is a yeast? And some were also modified from a previous instrument that was used. Some basic questions about their interest, just level of Likert scale interest of STEM and biology careers, and their confidence with 5 different scientific practices. Again, this was modified from a previous instrument that was used. So we did use a variety of data collection methods, some pre-post surveys, as well as some student interviews, and then some teacher interviews as well. And I want to emphasize that this was not intended to be like a large, robust evaluation of impacts, because this was an iterative development process. It was really important that we get feedback along the way, and we would modify things as we go. So just to keep that in mind as I talk through some of this. And we analyzed any of those open-ended questions with either qualitative content analysis or a scoring method that I'll talk very briefly about, as well as some of the short answer questions were fairly easy to evaluate. And interviews did focus more on some of the areas that we saw in the surveys that needed further clarity. And also sometimes they were a year later, they were post interviews to see what students were retaining and what they found interesting and were recalling from the experience. So I'm going to walk through kind of 3 cycles of data collection. The first year, as has been described, was with a California school, and he had actually 3 classes that were involved. So we did test out some of the survey questions with this class. And I won't say too much about this because obviously it was a sort of just a pilot to see how the survey questions behaved with this new population. And I'm noticing there's some chat in there. I just want to double check that. Okay.
The chat is about my talk. Sorry.
Okay, no worries.
Follow-up questions from that.
So we did want to see what students were thinking they liked or disliked from the activities. And as you'll see, they liked a lot of aspects of it. They liked watching the yeast change or evolve. Sometimes they used more specific terminology, and determining that amount of fungicure to actually increase. That was a really big piece that they brought up on their own when we asked about likes and dislikes. And seeing that mutation data was pretty interesting, as well as competing. The competition was pretty big on the popularity. They just didn't like losing. So that was one of the few dislikes. They didn't like to lose in the competition part of it. And some of them did talk about it being sort of repetitious or tedious, whereas others would mention, it became routine and we got good at that. We were able to practice scientific procedures. So although it might feel tedious, it's sort of obvious for anyone who is a scientist that is part of the process of doing science. And then we identified some confusion when analyzing the sequence data. And in turn, we're able to scaffold that a bit more in the later module usage. So that was a really helpful thing to hear. And all but one student agreed they would do it again. One was uncertain, and all of them enjoyed participating. So I think overall, we got some pretty positive responses. And I liked this quote sort of mentioning how it was fun to feel like they were doing real science, and even seeing this was an AP course, so seeing how the AP exam, it was really helpful to have actually done those processes rather than just memorizing terms. So again, emphasizing that students were finding this helpful, and we could address some of the challenges or confusions that they were having. So in the second year, we did have another school that was involved. And we didn't do as much with that data. I won't present very much on that. Just wanted to mention that one of the other questions that we were looking at, sort of emphasizing the importance of knowing prior knowledge for students, we asked in advance, what is yeast? And because you think the whole thing's focused on yeast, it might be good to know what they're thinking it is. And a lot of them, it's a very open-ended question. So just asked, a lot of them identified things like it was used to make bread or alcohol. And there were others, like they thought it was a fungus or a single-celled organism or bacteria. So certainly some variation in the prior knowledge coming in about yeast, just as an example of some of the things that we used earlier in the survey and dropped for later. Once we knew some of the things they were going to come up with, we could modify the lessons appropriately. So I'll just spend a little bit of the time. There's so much data from the 2019-2020 Idaho school classrooms, and I'm just going to touch on a couple things, for time's sake today. It's obviously to integrate all this into one analysis is impossible because there's so many different considerations across each of the years. So I'll just, focus in mostly on that Idaho classrooms and talk a little bit about some of the highlights. And just briefly, so you know what kinds of students that we're talking about here, this is across all three of the classes or teachers that we worked with. You can see that, there was some diverse populations here. Most of the students were around 15 or 16 years old. Most of them spoke English as their primary language. And one of the schools, obviously, as you can see, was an all-girls school. So focusing on those 2019-2020 Idaho classrooms, I'll just go through a couple pieces about the disciplinary content knowledge questions. We had 4 open-ended questions that we went through. Actually, I really need to give credit to the undergrad, Kristen Gunkelman, who went through and coded these questions for different types of codes that we thought were important to look through. So I'll give one example of that. The question was, what role do mutations play in evolution? And these were some of the student responses. She went through and coded these with a blind pre-post. She couldn't tell which ones were pre and which ones were post. And then we assigned a code to each of those. So you can see some of those examples here. And I'll just show you on the next slide is a graph of those changes in the number of codes that were used by students pre and post. So the pre, you can see on the x-axis walks through the different codes. The y-axis has the percentage of students who use that particular code. They could use more than one. And you'll see, we thought it was pretty interesting, the change, pretty dramatic change in variation from pre to post. This is across all of the Idaho classrooms, all 3 Idaho classrooms. So when they were talking about mutations and evolution, they were using terms around variation or some sort of description that we coded as variation. So that was a pretty interesting response. And you'll see, there's lots of rich data here to maybe dig into more, but that was one I wanted to point out, sort of an interesting one. And besides coding responses, we did score a few that we felt were better used as a, how correct the answer was. So again, Kristen went through and coded all of these from a 0 to 3 scale, from 0 being they didn't know, or it was completely inaccurate, to 3 being scientifically accurate and explained the concept. In this case, the concept was about how would you explain antibiotic resistance? So this is one of those applied questions that we were trying to see how it might compare to previous work, previous instruments usage. And I'll just very briefly, I'm not going to go into a lot of detail here, but you can see that increases here, I forgot to put which were statistically significant, but there were increases across the board, but obviously you can see there's some higher than others in this case. And we definitely struggled with getting some student responses in some classes. So it was hard to get some good stats on some of these, but overall average increase across for this particular question. In terms of their interest in STEM and biology careers, that increased on average for both. So that was great to see. And their confidence in science practices also increased. So I'll just show one of the 5 statements that they responded to, and you can just see the spread, again, pre and post across the Idaho classes in 2019-2020. So on the x-axis, you have from strongly agree to strongly disagree, and the percentage of students on the y-axis that responded with that way. And you'll see just that increase in both, the increase in strongly agree and agree for both post, and the decrease in uncertainty, disagreement, and strongly disagree for the post as well. So again, an overall increase in their confidence in some of these science practices. And we did have, as I mentioned, those student and teacher interviews as well. I'm not going to have time to go into a lot of those. I just wanted to give you 2 quick quotes from students that were taken a year later from them having been involved. So one, again, emphasizing this like real observations and actually getting to observe yeast evolving. The other really emphasized to us some of the things we could be thinking about to change, feeling like the lab had no set end goal, just sort of petered out, we lost interest. So how could we maintain something that's a long-term experiment and keep that interest going when it is something that they're doing sort of tedious, but how do we connect it better to what the full picture of where the data are going, how the data are used by researchers. So again, lots of things that were helpful to hear from the students, and then from the teachers as well. They suggested things like a standardized data log that would help them in the classroom. So lots of appreciation for the students and teachers who went through some of these interviews with us. And some just final thoughts here. It was really useful to have these multiple methods of evaluation to kind of triangulate the impacts, and then of course iteratively improve the modules as we use these across classrooms. It certainly was difficult to isolate the impact from yEvo because it's a long-term activity. They're doing other things in the classroom, so there's not just a set, they did this one intervention pre-post. They're actually doing these experiments over the course of multiple weeks. So thinking about some ways that we can maybe capture impact of yEvo specifically versus just them being involved in their classroom activities generally. And I think a lot of this has been great baseline data to be thinking about future education investigations. There's some really rich pieces that came out of the open-ended questions that I think would be useful for further investigation. And the expansion piece, Maitreya brought this up to, how do we expand this for other teachers to use? And especially the things that's come up a lot with the teachers that we've talked with, how do we do this for teachers that don't have easy access to researchers? What does that look like from an equity perspective? How can we make this even easier for teachers to implement? Again, balancing that with those who are leading, the researchers leading it, and sort of capacity. So just some final thoughts there. And again, as Maitreya already mentioned, you can find out more, same slide there. It's 2 p.m. Friday, in case you didn't catch the time on Friday. We'd love to have you join us for the sandbox. And just, thanks to everybody who's been involved, and of course, all the students and the teachers and Beacon, because that was the only way I've gotten involved with this project, was actually through a Beacon grant. So greatly appreciate initiating that collaboration. And happy to take questions, open up the chat and see if there was something in there I missed.
Yeah, Anya's got one about how this fits in with all the other things that the teachers have to teach.
Yeah, that has been a really big part of the questions from the teacher side of things. And the first teacher, so Ryan, usually talks about it's pretty easy to incorporate because they were able to do just a couple minutes at the 10 or 15 minutes at the beginning of class, and students could just go and do their transfers right there. There wasn't a lot of, he didn't need to lead them through it every time. They could take their own agency and independence to do that themselves. So that was something, he wasn't finding terribly difficult to incorporate. Now, other teachers and changes in the timing of classes certainly could have an impact on that. So that's, in part, that's a challenge. And I saw, Anya, your question about, if it's something that they do after the AP exam, that could be an option, I think, in part because this is intended to be multiple weeks. It really would be good for them to have that amount of time to work through it. And, they only did one module. Of course, last year they were interrupted by COVID as well for one of the classrooms, which made this a little bit more difficult. But it was mostly before AP exam. And so they were able to apply what they learned on the AP exam, which is kind of cool as well.
Rich, you're muted.
Yep, sorry, thanks. So I came in the middle of this because I'm jumping around between sessions, so I apologize, but I did catch, I heard Maitreya's talk, and then at the end you said students felt like it sometimes ended abruptly. And what about like having it being a multi-year project where students work with the material that other students evolved in previous years? You could actually even build some historical contingencies into it. But I think students, at least the really galvanized students, you could imagine that it would keep them engaged. Maybe once they're in college, what happened to their lines that now other classrooms are using. So anyhow, just food for thought.
Yeah, that's a great idea. And in part, I have to look back at where that quote, which classroom that quote was from, because it could have been COVID induced as well from that interview too. But yeah, I do think there's some great ways to keep this across classrooms. These were sort of 15, 16-year-olds, but if we could catch them earlier, if they're doing some other biology classes and get them started, yeah, that would be awesome. I love those ideas. And I'll lean on the yEvo lab group to implement them. But
well, and Rich, you'll like this result, but we had one class that decided to go the whole year. They were just going to evolve for 7 weeks initially, and they ended up going for the whole year. And some of the later strains have more mutations, and that's where we were starting to get to see the patterns of epistasis more. So there's definitely a lot of benefit in keeping them going.
Long-term yEvo.
Yes, long-term yEvo. Robert, you're next.
Yeah, nice, Alexa. It's fun. One of the things that made me think about is the kind of thing that I'm interested in now, which is trying to get across not just the practices, but really the values, the virtues of scientists and so on. And you mentioned sort of the feeling, at least some of them, of this being tedious and so on. But it's, in a way, you might think of this as an opportunity, right, to get across what comes up in our interviews as really important for science, which is perseverance, right? You've got to work through, a lot of the practice of observation takes a lot of work, and that's part of what you're buying into, and you have to develop that. So there might be a sense in which it gives you the opportunity to talk about, hey, this is actually something that scientists, have to push through, and that's part of the, in a way, the difficulty, but also the joy of sort of seeing something no one else saw because they really did persevere. And it's kind of the equivalent of coaching people in athletics, right? You have to work harder. You've got to overcome the tedium. Anyway, I just wondered if you had any thoughts about that and other virtues here. Specifically, I was wondering whether the stimulated curiosity, because that's really the core virtue that you want to get across. Did it lead people to say, hey, I wonder what, because that's another thing we want to try to cultivate.
Yeah, those are some really great points. I think for the first one, I would love to think more on maybe a part of a lesson. We really did let the teachers do as they would with the classroom activities. So other than having the regular data collection processes, I don't think there was as much guidance for teachers on what else they needed to cover. So that could be something we could include as a way to respond to students saying, hey, this is really tedious. Why am I doing this? So I think that's a great idea to maybe have that as a piece of a lesson that could go with those modules as well. To the other question, which you might have to repeat just because once I started talking, I forgot what the other part that you said.
Can you talk about how it stimulated curiosity?
Thank you. Yeah. So one of the pieces that we heard from some students, or maybe from the teachers more so, was that because it still felt a little bit like they weren't generating the question itself, that could be a side, sort of a secondary piece. Like, well, you've learned how to do this. What other questions do you have that then we could investigate? And that actually, I'm pretty sure it was one of the teachers in Idaho who was asking, well, what could I give them to then have them ask their own questions? So yeah, I absolutely agree that that's another piece that we could expand on this and really make it something that they're diving in on the curiosity piece, because that is a struggle with a lot of these cures, is that the question's already established and students are just filling out the data sheets basically. So how can we make that?
Yeah, I think that's a great way to do it. Often you're not in a position to really even know a question to ask until you've done something.
Yep.
But if you have done it, part of what that does for scientists is that opens another question, and right, that's how curiosity starts. So really one could build this in as sort of a little follow-up supplementary part, right? You've done this, what would be another question to ask, right? And anyway, there are ways of building this in. I think this has lots of opportunities for doing that. So very cool. Thanks.
Yeah, thanks for the questions. And I know we're running a little over the
there are only 3 talks in this session,
okay.
So I've dispensed with the time limits.
Okay.
Although there are people from other.
There are people jumping back and forth. That's true. I maybe shouldn't be so fast and loose, but okay. Well, we'll go ahead and move on then to Benjamin Person is the last speaker in our session.
Hi. So, you all see that okay?
Yes.
Awesome. So hi, everybody. I'm Ben Person. I am a senior undergraduate student here at Michigan State. I'm studying computer science, experience architecture, and game design development. Since fall 2020, I've been working on a project with Beacon, MSU Beacon, called Salmon Run, which is an educational game. And I wanted to kind of take this time to essentially go through some of our process, go through what we've been working through recently, and use it to contextualize how we can use games as an educational tool to kind of like help along with more traditional lesson plans, and explore a different avenue for teaching students. So real quick about Salmon Run. Salmon Run is a top-down 3D educational tower defense game. The idea is that every wave of the game, like a tower defense game, salmon are running upriver to go back to spawn. And essentially the player is contextualized as essentially an ecosystem manager of some type, who is managing anglers, rangers, setting up dams and other wildlife tools, salmon ladders, etc., to try to manage the population of salmon to make sure it doesn't balloon out of control or it doesn't go extinct, because both those things would be pretty bad. And I think like this has kind of been our focus to kind of teach, over wave after wave after wave, to teach, genetic drift, Punnett squares, dominant recessive genes. If we wipe out all the small fish, what's going to happen in terms of our proportions of medium and long fish, and that kind of thing. And at this point, it's probably worth noting, I imagine the vast majority of people in here are probably not super involved in game development projects. And so a lot of people end up usually asking me, why do we bother with games at all? Games have become, this multi-billion-dollar industry at large, but the vast majority of them are focused on entertainment rather than on education. And so people generally associate them with purely being, a thing done for enjoyment, a thing done for, that you do in your free time, not something you do with your schoolwork. And I think the big thing to take away is that a lot of what makes a good lesson plan and what makes a good game, in my opinion, often overlap. First and foremost, good lesson plans usually are trying to get the main points of a topic across to the students, efficiently, concisely, and a good game is trying to do the same thing, but with its mechanics. So the way you're playing a game, you're trying to make sure your player quickly understands what's going on, how the game is played, and what they're supposed to do. A good lesson plan also needs to be memorable. Same thing with games. We want students to remember the lessons that we're teaching them. We want them to be able to, study for their test. We want players who are playing our games to remember what's going on, so that they care, so that they're invested. We want it to be memorable, so they come and play it again and again. And then also, good lesson plans need to be fun. I think a lot of people here would probably agree that evolution is cool. Evolution is cool to learn about. Science is fun. That kind of thing. Learning is fun. And so naturally, we want people who are learning to take interest in the topic and want to come back for another lesson. And it's the same thing with a good game. If your game is fun, people are going to come back for more. You make a sequel, people are going to come back for more. So with all that kind of setup out of the way, I essentially want to go through a number of principles that I've been using as I've been working on programming for Salmon Run, for how we've been going about setting up Salmon Run and how we're framing it and how we go about designing it. So the first thing is that games are learning through doing. Everyone, I think, has mostly seen level 1-1 of Mario. It's got a couple of question mark blocks. It's got a Goomba. All you're doing in level 1-1 of Mario is you're walking along, you're jumping, and you're trying to get to the flag at the end of the level. I've played through level 1-1 of Mario with a number of people, friends and family, who have never played games before. And all of them generally do the same thing, which is they will hold right and they will run to the Goomba and immediately die. At which point they all say, "Oh, that thing kills me." And I go, yeah, it does. And what that is the gears have turned in their brain that they've associated running into an enemy with dying and losing a life. And all games essentially work on this fundamental principle that you're learning through your mistakes and you're learning through doing stuff and actually engaging with the mechanics at hand. All of Mario is just running and jumping. And it's not all that complicated, but you don't know how those things interact with each other until you do them. And it's also worth noting here that level 1-1 of Mario is pretty friendly. It's simple. It's straightforward. There's not a lot going on. There's a reason it looks like this and not the nightmare that someone made of in Mario Maker on the right that I don't even want to play through. I play tons of games. For Salmon Run, what this means is that we try to make sure that the mechanics of our game, placing anglers, placing rangers, placing dams, placing salmon ladders, also demonstrate what goes on if you place those things in an actual, ecosystem. So if you place a dam and you don't place a salmon ladder and there's nowhere for the salmon to get around, all the salmon are going to die, or at least get, the population is going to take a massive hit such they can't recover. And when, students have run through the game and seen that happen, that's been their reaction. They're not just learning that, hey, if I place a dam, all the fish die. They're also learning, oh, in real life, if human action isn't, met with other balancing factors, you're going to wipe out a population. The population's just going to die. The second thing is games require abstraction. Games are inherently very complex, even if you only have a few mechanics, and it's usually better to try and focus on a handful of mechanics. And because of that, it's very tempting to try to, since we want something to be teaching a lesson about evolution, we want it to be comprehensive, we want to account for all the factors. But doing that often adds a lot of complexity. One of the things that we were going through while we were making Salmon Run was figuring out how to make the fish path to the end of a level. And we started out by doing it via having the river literally apply forces to the fish that would eddy them about like they're being eddied by the currents. The issue is that's very performance intensive. Your frame rate tanks a lot. And on top of that, it also is not really about evolution. At that point, we're talking about the way that fluid mechanics work. That's not necessarily about evolution. So at that point, we just decided to simplify it, and we basically drew a couple of paths through the river that a salmon will randomly follow, and they'll just do it. It looks just as good, if not a little better than the first time we did it, and it focuses on the matter at hand and gives us more time to focus on other stuff that's more meaningful, that should have more performance-heavy stuff associated with it. 3, and this is a big one for me, is games are not a substitute. Back in freshman year, I worked on a research paper through the MSU Honors College. The physics department at MSU had made a game called Kirchhoff's Revenge, which was aimed to teach Kirchhoff's laws of electrical circuits to students. Essentially, they were trying to see if they could teach these laws to people who had no idea about what they were at all, myself included. So my job was essentially to help people out with playing the game, signing them in, getting consent forms, all that kind of stuff. One student in particular was kind of off in the corner, trying her best to play the game. She'd taken the pre-test, she was going through it. And I walked over because she waved me over, and she said, I think I have to go. I don't think this is for me. I'm getting a headache. I said, oh, absolutely, understand. Please, thank you for coming. We really appreciate your time. And she walked out. And I looked at her screen. She hadn't gotten out of the first room. And this is not necessarily her fault. She said when she had walked in, she hadn't played a lot of games and was really nervous. And despite all our reassurances and I was trying to help, she wasn't set up for success in terms of being game literate, so to speak. And that's true of a lot of people, not just students, but my parents aren't very game literate. It's not a medium that they interact with a lot, so they don't, they're not familiar with holding a controller. They get nervous when I hand them a controller. And so I think the important thing to remember for educational games is they're an auxiliary tool that will work for a lot of students who are familiar with games, who really love interactive media, but it's not necessarily going to be for everyone. It's something to add on to a lesson, not something to teach the lesson itself. And lastly, games are fun. I hammered this point home earlier, but I feel like it's very much worth reiterating. Back in elementary school, I used to play a game after I finished my lessons in the library that was installed on the computers called Type to Learn. The whole point of the game was you type sentences as fast as you can, get points based off how fast you typed, is to help you type faster. On the right here, you can see a different game that you can Google right away, and you'll find it in 2 seconds, called Type Racer. It's the same thing, and you just type out as fast as you can. Whoever finishes the sentence first wins. And for my part, I've always loved thinking back to playing Type to Learn more than I've played Type Racer hundreds of times, with friends who asked me to do it, but it doesn't stick with me. I don't remember anything about Type Racer, but I remember Type to Learn. And I remember Type to Learn because Type to Learn is fun, because I'm not just typing sentences, I'm typing sentences to help Space Gandalf in a Ferrari, save the Earth, which is wonderful. And so I remember that, and I remember, oh yeah, I have to keep my hands on ASDF and JKL semicolon, and that stayed with me. For Salmon Run, we're trying to do the same thing. We go ahead and we've essentially contextualized it as giving the player a role to enact. So they are the manager of this ecosystem. They are a wildlife manager of some variety. They're filling out policies and paperwork, which we obviously streamline as button clicks and such, but makes it more engaging than just, please drag in, the X, and the X will represent angler, and, we'll have a bunch of arrows, going down the river. It makes it more engaging. It makes it more memorable, and making the UI and everything fit that theming makes it more fun. All in all, to summarize, just kind of the big takeaways, that if you only remember one thing, remember this slide, is one, games are another option of learning tools to contextualize your lessons. I don't think that they fit teaching the lesson themselves, simply because of how many people don't jive with them because it's not their cup of tea. But there's something that you can hand to a student to, cement lessons firmer. The second thing, the way games are presented can heavily influence how a student responds to them. I think that giving them a role, giving them a story, giving them a narrative that is solid makes it more memorable, because once they have an actual thing in their head that is, in our case, we use real rivers. So the Columbia River is one level. Columbia River from the northwest of the United States is one of the levels. And when students can point to a map and say, oh, I know that river, it's right there, I can point to it, and that's, what I played on, they internalize what's going on more. Third, some of the best games are very targeted with only a few mechanics. The Super Mario principle, all Mario is moving right and jumping. That's pretty much the whole game. Your game doesn't have to be complicated to be fun and enjoyable and memorable. In our case, our game is mostly just dragging towers into the level. And if you're interested in making an educational game, or just exploring the ideas that you have in your head, I would say don't worry about originality. All good artists steal. They don't plagiarize, but they steal. And by which I mean, everyone has all the original ideas out there as far as genre and mechanic ideas have mostly been, discovered and dug up at this point. And most things are variations on a theme. So take what you have in mind for a game and try to run with that, and don't be scared if you're saying, well, this is kind of like, this game. We take a bunch of inspiration from a bunch of different games for Salmon Run. So just go crazy and see what you come up with. That was a lot to throw at you guys in 12 minutes, especially since I know it's something that I don't think everyone here is necessarily like specializing in. So questions, thank you for your time. I really appreciate it.
Awesome, that looks so fun. I have to ask, what are the upgrades you can buy at the upgrade store?
Right now, we're rethinking the upgrade system kind of entirely. We're going through and we're thinking about having anglers being able to get better with time. So the more fish that they fish up, the more effectively they're going to grab bigger fish, and the faster they're going to be able to, recast and attempt to grab more fish. For other things, we're thinking of systems like, initially you can buy a salmon ladder that is of one type, that's like a ramp, but you can also purchase, some what we would call side grades, which aren't necessarily better, but are focused on different kinds of salmon. So a ramp might help small salmon get up the best, but a salmon elevator that you can purchase and switch out might be better at helping a larger fish get through, and so on and so forth. So it's that kind of thing. Right now, we kind of have a handful of towers in. We're focusing on trying to get them working, and then we're going to think about how we upgrade them.
Other questions? Bryce.
I see a hand raised, yeah.
Hey, I was wondering during development if you've had any instances where a player was going through and something about the way the gameplay was designed gave them either a surprising insight that you were excited about, or left them with a misconception that you felt like you needed to design around as a result.
Based off where we're at, we've handed this off to a classroom or two. And kind of gotten initial feedback on what we've been working on. I don't think there's anything in terms of like really hard misconceptions that we've had, but I'd say the interesting that's happened is that handing it to a bunch of students in a middle school classroom, they tend to, and this is true of all games and testing all games and handing off to players, they will immediately find ways to either like break your systems in ways that you could not even dream of. Like it boggles your mind, or they'll come up with like ways of playing it that aren't the intended way to play, but are interesting. In particular, students would often take the game and they would say, okay, how fast can I wipe out the population? Which is like, oh, well, I mean, it's educational. It teaches you about, what things are really, really harmful if you, do them in excess. So they're still learning. It's just, that's not how I was, it's not how I intended them to do it.
being here in Seattle, I'm immediately thinking of an orca add-on where you can try to maximize the orca population and, the balance is there and things like that. So I love the idea of using local rivers to give students like a natural like familiarity point.
Yeah, and as far as like animals are concerned, like hunting animals, we have it so like if a dam is placed, a few turns later, sea lions will show up and they're starting to impact it. So now that's not necessarily something that you even accounted for. You placed a dam, you put a ladder, and it's like, great, we're done. No, there's all these other factors that are coming in. So there's that kind of stuff too.
Ben, you could tell them a little, since we have more time, you could tell them a little bit about the storyline that you and Sam have been planning.
So one of the things that is kind of in the future of our development is now that we have a lot of the mechanics in place, we've been thinking about, like I said, how do you get people in? What is the space Gandalf in a Ferrari for our game? And for us, we took inspiration from a rather dark game called Papers, Please, which is about essentially working a border post and stamping passports to let people in or reject them from entering an authoritarian country, which is very grim. But the thing that we took inspiration from was every day in that game, public events will happen such that, there was a terrorist attack, down a different border post, so you have to institute new rules for letting people in or not. Or, someone in particular might be passing through the border today that people want you to let through, even though it's not technically, per the rules. And we took that, we've essentially started making a storyline around for each river level that we intend to make. Have a series of events that will happen from wave to wave, from generation to generation of salmon, where you might have to account for certain stakeholders who are interested in keeping track of that river for different reasons, wanting you to do certain things. Maybe people, developers want you to put a dam in, but environmentalists don't want you to because they want you to let the salmon just be as they are. Which both have their pros and cons in terms of the results therein. Maybe there's suddenly a company that's polluting down the river that now you have to account for because salmon are dying at that part of the river due to pollution. So those kinds of random events and having that happen in a consistent fashion every time someone plays is kind of what we're shooting towards right now. So there's more of a story about this is what people actually deal with when they're trying to manage these ecosystems.
All right, anyone else? Super, this was a great session. I really enjoyed it, and sorry to any of you who were trying to hop between sessions, but I really enjoyed the extra conversation we were able to have about it. So hope to see you all at later events in the Congress, and thanks again for coming. And Louise.